make sure I record this. So those of you tuning in from home, read Poland chapter 1, and then, so here's the Moodle site for the course. I'm going to switch my view to what you can see, because I don't have everything available to you. So we haven't really talked about the Moodle site very much, but this is Moodle site that I rolled over from last time I taught this class. So I'm changing the dates and stuff as we go. But for example, for September 3rd, if you look in September 3rd materials, it has PowerPoints on basic plant structure. So this first full chapter of Poland's book actually involves a lot of plant biology. And so in order to understand that chapter fully, we need to know some things about plants. So we're going to be spending a lot of time here at the beginning on plant biology, which is not my favorite topic in the world, but it's just necessary. I'm an animal biologist, so plants aren't necessarily my thing, but in order to understand a lot of what Poland's talking about in chapter 1, which is the densest chapter of all the chapters of the book, you need to know some things about basic plant structure. Here are the instructions for the first food blogging. So go on to WordPress, and I'll go and show you the WordPress site here in a minute. Week 1 is this week, week 2 is next week. This week you're only required to post once, but you need to respond to 2 of your colleagues' posts. And so we're starting off easy on food blogging, just kind of get you into the habit of it. What was your favorite dish when you were growing up? Was it a special occasion dish or just one that you really liked? What was it about it that made you like it so much? Was it linked to a particular event? Et cetera, et cetera. So kind of think about food as you were growing up and sort of unpack that. Then the next week, I want you to concentrate on thinking about what you're eating. We are going to be starting to talk about plants a lot, and the basis of our food chains that we're involved in. We'll talk about food chains here in a few minutes when we talk about the introduction of Poland's book. Plants are the base. Either we eat plants or we eat other animals that ate plants. And so thinking about the mix of our diet, how much of our diet is meat versus plants, et cetera, all of these things matter when you start thinking carefully about food. So these are the instructions. This is what I want you to be commenting on. And then I want you to be commenting on one another's posts as well. The WordPress site is called The Ecology of Food. And when you go to view The Ecology of Food, what you're going to see is nothing found, because none of you have posted anything, because I just invited you to the site. One of the things that you can do is you can go to the post section here, and you can start writing a post. So I've started making my own blog post, and for me, it's going to be about my favorite dish. So I started writing it here. Let's see if I can make this bigger. Oh yeah. So my favorite dish doesn't come from being a child, it comes from being an adult. It's chicken mole. Have you guys ever had chicken mole? Oh really, where?
Just like one of my parents' friends from Mexico just made it. Yeah.
Very fortunate to have friends from Mexico. Mole, do you know anything about mole?
It's made out of chocolate.
Yeah, it's billed as chicken and chocolate sauce, but that's really a kind of misrepresentation because it's mostly chiles. And so I started writing this up this morning, and then I have some photos on my phone from the last time I made mole. So I make mole from scratch. It takes me 2 days. 2 hard days of work. Has so many ingredients. And so I went to get the photos, and I was just going to show you photos, kind of walking you through the process in the blog post. And so this is the recipe card. The recipe card is 3 very dense recipe cards to make this one recipe. Let me just see if I can do it this way. and you have to gather all your ingredients together. You've got fresh vegetables, you've got dry goods, you've got seasonings, you've got a bunch of chiles. And then you saute all this stuff up. You roast the chiles, 3 different kinds of chiles. I now grow all of these chiles in my garden so that I can have dried chiles. When I would go and visit my family in Texas, which is where I'm from, whenever I was running low on chiles, I would go to the Fiesta food store in Conroe on my way back, and I would just buy up the necessary chiles and bring them back and put them in canisters in my kitchen so that I had them when I needed them. Because there's nothing worse than going to make some mole and then discovering that you're short some chiles. You cook all of the spices in lard. You cook all of this stuff in lard. You mix it all together. Then you put it in a blender and grind it up really fine. Then you, after the chiles have been roasted, you put them in hot water, and then it kind of steeps out the flavor of them. You put all that stuff in a blender. Then it kind of boils down to a paste. The chiles, when you blend them, they're kind of pasty, and then you cook that paste. You basically fry it in, once again, in lard. Everything is in lard. Everything's better in lard. Then you add some chocolate. So there's very little chocolate, actually, relative to the amount of other stuff that's in there. And the main flavor comes from the chiles. And then after you've cooked that for a while, you've added the chocolate there at the very end, added a little bit of sugar to whatever your level of sweetness is. Then you have to strain it through a fine sieve to get all that chunkiness of the bread and the tortillas and all that vegetable material. It gives it a really grainy texture. And the process of pushing it through a strainer is amazingly time consuming. You're just standing there at the sink, pushing this stuff through the strainer, and it gives you this glob of gritty, grainy stuff, and the mole that comes out at the bottom of the strainer is really nice and smooth. And then you make it into enchiladas, and it's very good, but it's like my favorite dish in the world, so I'm writing you a blog post kind of so you can see what I'm looking for about mole. You don't have to go to this level of elaboration in your blog post, but these are the kinds of things that I'm interested in. This is one of the ways that I get to know you as a human being to a certain degree, is through food. So anyway, I went to upload these photos, and they're all taken on my iPhone, and WordPress won't take iPhone photos, so I have to get them all converted to JPEG before I can finish the post. So anyway, so I was doing this, and I got to the upload portion of it, and I went to upload, and it's like, won't accept that file format. So I have to pause. But as you're doing this, if you're writing a post, if you're in the process of writing a post, you can go up here and you can save it as a draft. This is now saved as a draft. If it wasn't saved, it would give you an option up here to save it as a draft. And then you can come back to it and add to it later. And then when you're ready to publish, just click the publish thing. Now, when you publish, I want you to put your name on the post. So I'm still in the process of learning names. JT. So name your post things like this: JT Sherman, week 1. Then next week, when you post, do JT Sherman, week 2. And then we're going to start a new set of prompts, a new set of things that I want you to be talking about. That will then be week 3, week 4, week 5, week 6, week 7. Now you can add to this week 1 favorite foods if you want. But for me, as I'm grading this as part of your grading class, it's helpful to me if I have your name attached. If everybody names their blog post my favorite food, then I have to go through and figure out, I have to look at the author line once I get into the post to see it and stuff like that. Just it makes it a little easier for me to keep track of what's going on. So that's my only request on that. Other than that, it's kind of a free-for-all. Just respond to the prompts and write what comes out of you. When you're commenting with one another, be respectful. Don't say things like, chicken and chocolate sauce, that sounds disgusting. How could any human being eat that crap? I don't expect you guys to say anything like that. But when I first encountered mole, they billed it to me. I was going to interview at graduate school, and these graduate students took me to this Mexican place, and it was on Thursday, and Thursday was mole day. They only serve mole one day a week because it's so difficult to make, so time-consuming to make. They're like, oh, you should try the mole, it's great here. I'm like, what is mole? I didn't know what it was. I didn't have any friends from Mexico. And they're like, oh, it's chicken and chocolate sauce. And I'm like, ooh, I'm not sure that sounds great. And then I ate it because I was just open to trying new things, and it was excellent. And then I traveled extensively through Mexico all through graduate school for a variety of reasons. Mainly I was dating a linguist who worked in Mexico. And every place that I stopped, I would try the mole because there are regional differences throughout Mexico and Central America in mole. And one of the professors at the school that I ended up attending, which was the school where the mole was 2 miles away on every Thursday, he made a really careful study informal study of all the different varieties of mole throughout Mexico and Central America. He was a herpetologist and was from Guatemala and traveled extensively throughout Mexico and Central America. And so he was a great resource. Oh, Dr. Campbell, I'm going to this part of Mexico. Where are some good towns to sample mole in those areas? And he would tell me all about mole. You'd get him started on mole and he could just go for days. So anyway, it was a little off-putting. But once again, as soon as you taste it, you're like, oh, this isn't really chocolate. This is really chiles with just a hint of cocoa in there. So anyway, food blogging. Any questions about that? Okay. Once again, don't leave it until the very end of the assignment period, which is also why I will oftentimes break it up into 2 weeks. I want you to do this during week 1, and I want you to do this during week 2. That way you kind of space it out, gives other people time to respond to your posts, et cetera, et cetera. Like I said before, if everybody logs on Thursday before it's due on Friday and posts, there's no opportunity for people to comment on those posts because there's just not enough time. Questions? All right, so look for the email that you should have gotten from WordPress. It's not spam. It's actually for me asking you to join this blog. All right. Questions about what we talked about last time? We were talking about, if I recall correctly, at the very end, we were talking about sensory biases. We talked about how we have a sensory bias towards sweet things and an aversion to things that taste to us bitter. And I talked a little bit about the neurobiology of how we perceive things like tastes and smells and things like this. Those perceptions are interpretations that our brain makes of basically neural inputs that it's receiving. And so while something might taste bitter to us, it might not taste that way to some other species. It doesn't have that evolutionary history that has shaped their brains to view those particular stimuli as bitter. And so we have sensory biases towards things. We have a sensory bias towards red. I noticed that 2 of you are wearing red t-shirts today. The school colors of Williamsville College are red. The label on Coca-Cola is red. The label on Pepsi is blue, but you notice it has a red stripe in the middle of it, don't you? Well, Coca-Cola predated Pepsi. It came along before Pepsi was a thing. And if you're marketing something, you don't necessarily want to make your labeling similar to a competitor's labeling. You want to stand apart. But even within that change in labeling, not being red but being blue, there's actually a flash of red. When you look at When you look at signals in birds, something like a red-winged blackbird, it's this jet black bird with a little flash of red on its wing. That red is a sexual signal. It's a sexual signal that basically advertises male quality to female birds. It's like, hey, look at my red patches. I'm sexy. Lots of vertebrates have a enhanced visual response to the color red. Next time you go to the grocery store, and there's going to come a point in this class where one of the assignments during the week is going to be for you to go to a grocery store and map that grocery store. What's the layout of the grocery store? Where are the different kinds of foods located in the grocery store? Because the layout of a grocery store is not happenstance. They didn't just randomly throw stuff around in the grocery store. They put the milk and eggs in the very back, furthest away from the door. Why?
So you have to walk through, like, past everything.
You have to walk through the whole freaking store to get milk and eggs, because basically everybody gets milk and eggs. And so they force you to go through the whole store in order to get back there. The more time you spend walking those aisles, guess what? The more likely you are to buy something you didn't come there to buy. When you go to a grocery store, look at the packaging, and when you pay attention to it with a critical eye, you will be surprised at how much food is packaged in red packaging. Why? Because people who market food know that the human eye and the brain's interpretation of visual input is drawn to red things. That's why women wear blush and lipstick. The eye is drawn to red things. It's quite frankly why we have a mascot that is the Cardinals. More than likely. Certainly why cardinals are red. It's the male cardinal that's completely red. The female cardinal is a mixture of gray and red. That red coloration in cardinals is once again a sexual signal, because once again that catches the eye of the female cardinal. So we have these sensory biases, and they shape the food that we eat. You don't go to your refrigerator, whip out the broccoli container, and just sit there and munch on an entire 3 heads of broccoli. But you might go to the candy jar, get into the candy jar, find a couple of packs of M&Ms, and just sit there and pop M&Ms until the bags are gone. Because we have this sensory bias toward sugar, because we evolved under situations of food limitation. So that when we had easy to acquire sugary substances, high caloric substances, we ate all of those things because we didn't know when we were, our bodies didn't know when we were going to get that pulse of energy again. We evolved as hunter-gatherers eating high fiber diets, high protein diets, low carbohydrate diets. And so we have this kind of hardwired response to sugars that allows us to prefer sugars over other things when we encounter them. So, our biology kind of works against us in some ways. Paulin, in the process of kind of unpacking all of this, he refers to the French paradox. What's the French paradox? I've been talking a lot. Time for you guys to talk. Yeah.
They eat a lot of cheese and they don't die for some reason.
A country of people who seemingly eat in the most unhealthy way, eat the most unhealthy foods, and are just walking around healthy as can be. Paulin probably writes it better. Let me find this section. The French paradox, for how could a people who eat such demonstrably toxic substances as foie gras and triple creme cheese actually be slimmer and healthier than we are? So here you have a bunch of people, an entire nation of people, and I experienced this when I lived in Spain, an entire nation of people who basically eat stuff that we are told not to eat in the United States, and they're much healthier than we are. Lower rates of obesity, lower death rates due to things like heart disease, et cetera, et cetera. That's the French paradox. People eating unhealthy foods and are more healthy than we are. When I was in Spain, I lived in Sevilla. Walk out of your door in Sevilla, walk down any street, and at 10 o'clock in the morning, you will see people sitting around at cafes enjoying themselves. Paradoxical for so many reasons. At the time that I was there, unemployment rate in Spain was about 17%. And I asked myself, how can these people afford to be sitting around at cafes, eating food and drinking wine at 10 o'clock in the morning when there's 17% unemployment? But the other thing was, they were all pretty healthy. And like from 10 o'clock in the morning on, except for the siesta time, in Spain, you see these sidewalk cafes and they're just chock full of people. Entire families, intergenerational families. Mom, dad, children, grandparents sitting there, grandparents eating whatever they're eating, oftentimes what we would consider unhealthy foods, giving little sips of wine to their grandparents when their parents aren't watching, or to their grandchildren when their grandparents giving sips of wine to grandchildren when their parents aren't watching, et cetera, et cetera. You're just out there having a nice, good time, eating food that a dietitian in the United States would say you shouldn't be eating. And once again, you look at them and they're perfectly healthy adults. So we have this French paradox, but then he contrasts the French paradox with what? He calls it the American paradox. What's the American paradox? Yeah.
And unhealthy people obsessed with the idea of eating healthy.
A notably unhealthy people obsessed by the idea of eating healthy. So we as Americans look at the French and we're like, oh, what a paradox. They eat unhealthy, but they are healthy. But the French and everyone else in the world could look at the United States and say, there's an American paradox, a culture obsessed with eating a healthy diet who are among the most unhealthy people on the planet if you look at the culture as a whole. High rates of obesity, high rates of death due to things that are totally preventable, like cardiac disease, atherosclerosis, etc. High rates of diabetes. And so one of the purposes of Pollan's book is to kind of explore these things, explore the American paradox, explore how we got to the point that we are in terms of what food is available to us and why we have the dietary choices that we have and why we make the dietary choices that we do. So at the end of this chapter, he talks about these three food chains that he's going to follow throughout this book. What are those three food chains? Yeah.
There's the industrial food chain and the hunter-gatherer food chain.
So those are two of them. Industrial is what he tackles most and is what makes up the bulk of the book. there's one that he covers in the second part of the book, kind of in between industrial and hunter-gatherer.
Agricultural?
Well, no, because industrial is agricultural, it's just a weird industrial style of agriculture. And he refers to the hunter-gatherer as the personal food chain. It's a weird terminology, but that's what he calls it. But it's a hunter-gatherer, a treatment of thinking about hunter-gatherers. We have industrial, system based on corn and soybeans and other large commodity crops. Yeah.
Says the word organic.
Organic is actually a subset of the industrial. So there's something other than that.
Pastoral.
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 Yeah. And I should have brought my own markers because these are basically dead. And I forgot them in my office. So the industrial food chain is a food chain that is largely based on using heavy inputs of things like fertilizers, pesticides, et cetera. Machinery is used a lot in the industrial food system, tractors, combines, things like that. And so you could have part of the industrial food chain that is very conventional in that they use fertilizers and pesticides. But organic food, we're going to have a whole section where we kind of unpack what that labeling organic means. But the organic food chain is very much like the industrial food chain, except that some of the inputs are different. So for example, you can't use pesticides per se, unless that pesticide is a naturally based pesticide. And so you can't use fertilizers per se. You can't use fertilizers produced in a fertilizer plant, but you can use things like manure as fertilizer. And so the organic food chain is very similar to the more conventional industrial food chain in terms of the inputs that go into it. Just the nature of those inputs are different, not the quality of those inputs in a sense. The pastoral food chain is a food chain that largely tries to get away from those types of inputs. And so he'll talk a lot about this guy out in Virginia, a guy named Joel Salatin, who has this farm where he basically raises grass-fed beef, chicken, sheep, pigs, without essentially any inputs from outside the farm. Everything that the animals eat is grown on the farm, and he rotates those animals through his pastures in such a way that he's able to maintain productivity while also not depleting the resources of the farm. And that's the pastoral food chain. And then the last food chain is the hunter-gatherer, and this is generally the food chain that we don't spend a ton of time on in this class, simply because of time constraints. But these are the three food chains, and the first one, and the one where he spends most of the book, is the industrial. Then he spends a little less time on the pastoral, and he spends the least amount of time on the hunter-gatherer, partly because it's so divorced from what we currently do in the modern world. There are very few hunter-gatherer cultures still out there functioning as hunter-gatherer cultures. One of the things about being a hunter-gatherer is it requires large amounts of land to wander over in order to meet your needs, because as you forage in an area, you deplete resources in that area, you need to move on to another area. And so as private land ownership has become the norm across the world, the amount of area that hunter-gatherer cultures have available to them has shrunk. Not only that, these hunter-gatherers are oftentimes indigenous people, and they've actually been forcibly put on things like reservations. This happened in the United States with Native Americans. It happened in Australia with our Aboriginal people. It happened in Southern Africa with the Bantu people. They've been just increasingly put on smaller and smaller areas of land that are circumscribed, and most cases by the government, and that has made a hunter-gatherer lifestyle more difficult to achieve simply because you don't have the area that you need to hunt and gather over in order to survive. So this is basically how we all once were, but this is now a very rare occurrence. Very few people in the world actually exist as hunter-gatherers. So, any questions about the introduction? All right, I didn't give you any guide as to how to go about reading the introduction. But I'm going to give you a little bit of a guide as to how to read chapter 1. Here's the tricky thing about Paulin's book. You can read Paulin's book and think that you understand what he's talking about and not actually understand what he's talking about, because he's a decent writer, and if you read it superficially, you can read through it, you will skim over things that you don't quite understand, but at the end of it, you'll think that you have a pretty good idea of what he's talking about. And the fact of the matter is, when I read the first chapter of this book, I'm like, man, you have to know a lot of biology to get what he's coming from. So I'm going to clue you into some of the things that I want you to be paying attention to while you read chapter 1 in preparation for Friday, because they're the things that your brain want to gloss over, and when your brain wants to gloss over that, that's actually when you need to dig in deeper. So, the first part is actually pretty light, but he gets to this part in the second section of chapter 1, starts on page 19. The section is called Corn Walking. In this section of the book, he refers to corn walking, and when he refers to corn walking, he's actually referring to human beings. And he mentions that there's a guy who does what's called stable isotope analysis. And if we look at the isotopes that are in your body, we can get some insight into what you're eating, especially what you're eating relative to C3 versus C4 plants. And you're like, C3, C4 plants, I don't really know what that is. Do I really need to know what that is? Probably not. I'm just going to keep on reading. Maybe it will become clear. It doesn't ever become clear. So one of the things that we're going to be doing on Monday is we're going to be digging into photosynthesis, because only that way do you know what C3 and C4 photosynthesis is, and only when you know that does corn walking make any sense to you. So one of the things that you should do when you get to this section of the book and you see things like stable isotope analysis, you should go look up what the heck stable isotope analysis is. When you get to the section in that section of the book where he talks about C3 and C4 photosynthesis, you should endeavor to go and look it up and see what that is about. We're going to go into it in a great deal of detail, but in preparation for those discussions, if you could do a little bit of that work of figuring these things out beforehand, that will actually help us as we go through it in class. We are corn walking. If you go to Central America had a culture that was based on the cultivation of 3 crops: corn, beans, and squash. Sometimes referred to as the 3 sisters in Central American culture. Corn stalks would grow up, they harvested light, they're a C4 plant, as it turns out. Then the beans would grow, and as the beans grew, they would wrap themselves around the corn stalk and use the corn stalk for support. So rather than having to put artificial supports like trellises and things like that, you could just plant corn, plant beans next to the corn, and they would trellis up the corn plant. corn is a plant that likes a lot of nitrogen in the soil to grow effectively. Nowadays, we take care of that by putting out nitrogen fertilizers, anhydrous ammonia. If you go out, if you're driving through the countryside, in the spring in particular, you will see all these big tank trucks sitting out in the fields. Those are tanks of ammonium nitrate. And they pump that ammonium nitrate into the soil as a nitrogen supply for their corn, essentially. Corn needs a lot of nitrogen. Beans are a type of plant that take nitrogen out of the air that is not available to plants and convert it into nitrate that is available to plants. So when you plant your beans next to the corn, the beans benefit from the corn in that it gives them a place to grow, but the corn benefits from the beans because the beans put nitrogen into the soil. Squash, if you've ever grown squash, tends to vine out, has these big broad leaves. Plant squash at the base of your corn and bean plants. Those leaves basically spread out, produce not only squash, but also serve the purpose of shading out weeds and things like this. And so they would grow these three crops in combination with one another because each crop gave the people something to eat, but each crop also facilitated the success of the other adjacent crops. Pretty brilliant. But when you think about it, because they had a society based on corn, beans, and squash, their primary carbohydrate source was corn. Source of sugar or starch was corn. And so if you look at the stable isotopes of people from traditional cultures in Central America, look at the isotopes of carbon that they have in their bodies, it will show the signal of eating a lot of C4 photosynthetic plants. C4 plants are things like corn and sugarcane. Those are both C4 plants. C3 plants are beans and squash. And so you can see a carbon signature when you do a stable isotope analysis where these people have enriched levels of carbon of a particular isotope. And once again, we'll talk about that in detail. Turns out when you look at Americans, we have a higher isotope concentration of those isotopes that are fixed by C4 plants than people in Mesoamerica do who have a culture that is based on the use of corn. So we can look at our stable isotope ratios and see that we are people of the corn. That's a horror movie reference. And we're more so than people who have a traditional culture that is based on corn. And so we have an industrial culture that is also based on corn, and you can see that in this. So we have to understand some things about photosynthesis in order to understand this little section of the book. The rise of ZMAs basically begins talking about the rise of agriculture in Central America and Mexico. The evolutionary history of corn is involved in this. And also it talks about, let me make sure that I'm telling you correctly. It talks about kind of how, as corn changed over time, you can go on to the internet and search for images of corn. Corn started out as a plant called teosinte. That slowly evolved into corn over time. You can go and look at a picture of teosinte and all of the corn intermediates that have been taken out of middens from Central American and Mexican middens. We'll talk some about middens as we go through this. Middens are fascinating to me. But it talks about corn evolving from this small plant that had very few grains to these plants that have a lot of grains, a lot of productivity out of a single plant. And so that talks about sort of the evolutionary history of that. So we're going to be talking some about evolution over the course of the next couple of weeks. And then this idea of married to man, the next section, which is we could also say married to humans, because it's not just men who do this. This talks about corn reproduction and how corn would not be able to make it without the intervention of human beings. It wouldn't be a plant that could survive in the wild on its own and propagate itself on its own. And so we're going to have to talk some about corn reproduction. In order to do this, we need to know something about plant reproduction in general. So pay attention to those aspects of that section of the book. The next section is called corn sex. That's where the corn reproduction comes in. And when we talk about corn reproduction, plant reproduction, the section on corn sex deals with genetics. This is just chapter 1. Very dense chapter, a lot of science in the chapter. So what we're going to do is we're going to be walking through how to understand chapter 1 by basically covering a ton of science. So if you go to Moodle, go back to Moodle. Go to the syllabus and schedule, pull up the schedule. You will see that we have plant biology, plant biology, plant biology. This is photosynthesis, C3, C4 photosynthesis. We're a little behind, but we'll catch back up. We then have to talk about genetics. So basically, exam 1, an entire exam of material, is chapter 1 of this book. So read chapter 1 carefully, read chapter 1 in a way that you're extracting as much information as you can about it. Don't read chapter 1 passively. Be an active reader of chapter 1. I'm going to give you some chapters from a book called Plants and Society. Digitally, I decided not to have you buy the book because we don't cover all of the chapters in the book, and it's a really expensive book, and I didn't want to put that financial burden on you. I'll be posting those. I started to scan them today, but when I went to the scanner, another faculty member was using it, and I just didn't have time to get it done today. Last time I taught it, I had you buy both books, The Omnivore's Dilemma and this other book, and I decided to just stop doing that. Don't tell the people at the bookstore. So that's what's on slate for the next big chunk of September. For the next 20 days, we're going to be talking about chapter 1 in Pauline's book. And so that's where we're headed. But it's a lot of new material that you've never seen before. You might have run across some of these topics back in high school in a biology course that was required as part of your curriculum, but I'm guessing chances are you don't remember most of that. So when I teach you these things, I'm only teaching you what is necessary in order to understand what Pauline is talking about in the book. At times, you're going to think, man, that's a lot of really detailed biology. But for example, one of the things that Pauline will talk about in the book is genetically modified organisms. And so in order to talk about genetically modified organisms adequately, you have to have a knowledge of genetics at a pretty basic level, but like more than any of your friends know about genetics. that, but in order to understand the genetic modifications that he's talking about, and what we normally think of as genetically modified organisms, you need to know about the structure of DNA, and you need to know how DNA operates inside of a cell. I'm not teaching you those things because I think you guys are all going to convert to biology majors. I'm teaching you those things because in order to understand what a genetically modified organism is, you need to know what the types of genetic modifications are that are being made and how they're being made, and in order to understand that, you have to understand the basic structure of DNA and how DNA works in a cell. So, as we go through all of this, keep in mind that I'm being careful about what I'm asking you to know and why I'm asking you to know it. It's not just a bunch of science trivia that I'm giving you because I'm a scientist and I think science trivia is cool. It's information that I'm giving you because it really is integral to understanding the things that Holland is talking about. But it's also integral to understanding things like the COVID pandemic. So, and I didn't talk about this in this class before because we didn't have a pandemic. So, I got the Moderna vaccine. If you got your vaccine, you probably got it here at Jewel, I'm guessing, back in the spring. You also got the Moderna vaccine. The Moderna vaccine is a messenger RNA vaccine. mRNA. The little m stands for messenger RNA. So when you need to make a protein, you need to build some muscle or something. There are genes in your genome that code for making the proteins that are involved in making a muscle. There are a lot of proteins involved in making a muscle. So there are lots of genes that go into making a muscle. Well, your cells all have the same DNA in them. All throughout your body, every cell has the same DNA. The difference between a skin cell and a muscle cell isn't the DNA that's in them. What makes those things different is what genes in that DNA are being turned on or not during the development of that cell. And what we mean by being turned on is that the DNA molecule is triggered to make a messenger RNA molecule. And that messenger RNA molecule is the building instructions for building the protein. DNA is super stable. Hangs out in your nucleus for your entire life. Messenger RNA is transient. It doesn't stick around for very long. If you've ever tried to isolate messenger RNA in a lab, one of the problems with working with messenger RNA is that it breaks down really quickly. Well, why would messenger RNA why would a cell want messenger RNA to break down quickly? Take some guesses. Yeah.
So it can use it as soon as possible.
Well, so you want to use it, but then once it's used and it's made the protein that it needs to make, you don't necessarily want it hanging around any longer, because the longer it hangs around, the more copies of that protein it's going to continue to make. And so messenger RNA breaks down relatively quickly so that it's not used over and over and over and over again, because a cell might only need 1,000 copies of that protein, but if that messenger RNA hangs around, it's just going to keep on getting used and used and used by the cellular machinery that makes protein. So messenger RNA breaks down really quickly inside of cells, to the point that if you're trying to do something with it in the lab, you have to take all these precautions to keep it from breaking down. You have to use special tubes that don't have any contaminants in them that might interact with messenger RNA and break RNA down. So DNA is what's in your entire genome. Within the DNA, there are lots of different genes, and when you need a gene to act, it gets upregulated. It produces messenger RNA. Messenger RNA is the template that is used to make a protein, but then once those proteins are made, the messenger RNA breaks down so the cell doesn't continue making that protein beyond what's actually needed. So the Moderna vaccine and the Pfizer vaccine, for this matter, I'm not sure about the Johnson & Johnson vaccine. I don't remember if it's a messenger RNA vaccine or not. But the two most common ones and the two most effective ones basically have created, the scientists have basically used the genetic code of the virus to know what kind of messenger RNA to make. And so you can manufacture messenger RNA. And then when they inject this messenger RNA into you, your body's protein making machinery takes this messenger RNA and says, oh, there's messenger RNA floating around here. I must make a protein out of it. They don't think this. They're basically just little automatons inside of your cells. But because you have this messenger RNA hanging around, your body converts that messenger RNA to protein, and it makes a particular protein that is found on the outside of the virus. This thing is called the spike protein. And the spike protein is the protein that is responsible primarily for creating an immune response in your immune system. It signals cells to build antibodies to the outer coating on the outside of this virus. So when people heard that they were putting messenger RNA into people's bodies, what were some of the things you heard about people objecting to getting vaccinated, especially with regards to it being a messenger RNA virus? A lot of people were fearful of the vaccine because of this. You guys don't listen to the news at all? I know you do listen to the news because I've seen you on your response cards tell me where you get your news from. What's one of the reasons people were fearful of the vaccine? Related to this conversation. Yeah.
There's no virus in the vaccine, which is pretty common, right?
So most vaccines actually have the virus in there. It's an inactivated form of the virus. But in this case, they were putting messenger RNA in the vaccine. And people were worried, oh, you're putting viral genes inside of me. Isn't that going to do genetic damage to my cells? But the fact of the matter is it's not viral DNA. It's not even a full virus. It's just messenger RNA. And messenger RNA, it's only the section of the genome that produces this one protein. It's not the full genome of the virus, so you couldn't even create a whole virus from it. Not only that, messenger RNA breaks down relatively quickly, which is why they have to transport it in these cold containers, which has been a problem because if you don't have the appropriate freezer, et cetera, you can't store it appropriately, and it breaks down while it's waiting to be injected into somebody's arm. So as opposed to things like measles, mumps, rubella, those types of vaccines where you actually are getting a whole viral particle that's just been deactivated, in this case you're just getting a little snippet of messenger RNA that is designed to produce the protein complex on the outside of the virus that then forces your body to mount an immune response. As a result, in this way, it should be safer than the regular vaccines for other diseases because you're not getting a whole viral particle, even though it's been deactivated. You're just getting a little snippet of messenger RNA, and that messenger RNA breaks down relatively quickly. It's in your body just long enough to elicit an immune response, and then it breaks down like all messenger RNA does. A very unstable molecule in terms of holding itself together over time. So people were all up in arms about this, but ultimately, they're all up in arms about it because they actually don't understand, and they don't know about, and they've never been educated about this process of moving from DNA to messenger RNA to protein production. This thing in biology is called the Central Dogma. It's a fundamental idea in biology, and it's relevant to thinking about vaccines but it's also relevant to thinking about genetically modifying plants. So you're going to understand what I've just told you about vaccines at a much deeper level after we go through the Central Dogma as I'm gearing you up to talk about genetic modification in plants. So little did I know when I formed this class that there would come a point where there are parts of the class that would be relevant to you in ways not related to food. But here we are in the year 2021. Questions? All right, we're done for today. I need to talk to you guys before you leave. Read chapter 1 of the Omnivores 11-month Flag Friday. And look for a posting of the Plants and Society chapter as well.